This is the Notability review notes that I was going through with you this week, and I thought it might be helpful to look at them again in a video format. And this is also for those students who may have missed this. So I gave these notes out, and you have them, at least parts of them that we added some annotations on this, as a way of sort of organizing this chapter. So we've looked at several aspects of metabolism and enzymes. And the first one was just, what are they? What are enzymes and what is their role in metabolism? So I gave this as a suggested review for your own study purposes, to think about this as maybe a free response type of question, where you think about some enzyme, and here we have an example here. I gave sucrase and a substrate, which would be sucrose, and looking at it in terms of like, well, what does sucrase do to help, in this case, break down sucrose? So we can see this over here on this graph, where you see that sucrose has some free energy available. So that's right here, and that we can break down sucrose to get some products from it, and those products would be glucose and fructose, and release energy, some energy in the process. So when this happens, this reaction happens much more readily when the enzyme sucrase is involved. And the reason being is that sucrase, is going to take sucrose, remember, into its active site, and it's going to create a situation where it's more easily broken down. So very often it gives it that induced fit. It kind of snuggles around its substrate and helps it to break down into these products. So this is an exergonic reaction. It's a catabolic reaction. We're breaking stuff down. We're releasing energy as part of the product side. And just to remind you of what this is, this increase, notice that we have two things here where we only had one thing here. So we've kind of increased disorder. We've increased entropy. And I'll go on to Gibbs free energy a little bit later. So this is just one way to look at it. Remember that there are some models that we use. One is that induced fit model. One is a lock and key model that kind of explains what enzymes and substrates kind of do. The idea of the different types of reactions, one of them I just mentioned above, this catabolic reaction or exergonic reaction. So kind of knowing some of this vocabulary is helpful. And then Gibbs free energy I'm going to talk about later. So I've mentioned it before, but we'll get into a little bit more specifics. So that was one thing to do for review. Part two, what things are going to affect how enzymes function? So looking at it from the environmental standpoint, and I mean by environmental, I'm talking about inside your body or inside an organism's body. So we know that enzymes are very specific in terms of their shape, right? They're proteins, and so shape is really important, and that certain things can affect their shape, and that would be temperature, concentration. Well, actually, sorry, this doesn't affect their shape. This is just another factor. pH would affect their shape. So temperature and pH would be the two main ones that could change the shape of an enzyme. But there are other things that can affect how enzymes function. One of them has to do with what's the availability of a substrate. The other would be things which we call cofactors and coenzymes. So these are things such as the one you see here. This is from your OpenStax reading. This says, oh, yeah, vitamins like vitamin A or vitamin B, some of these are necessary for enzymes to function. So they support the enzyme, they enter the structure and stabilize an enzyme. They do all sorts of things. So those would be your vitamins. These would act as coenzymes. And when we say that these are coenzymes, we're looking at something that's really an organic structure here, right? Lots of carbons and hydrogens and oxygens, etc. That's what our vitamins are made up from. The other thing that can be useful for enzymes and sometimes critical for enzymes to function are these things called cofactors. So these are what you'd say are the minerals, right? These are the elements or the ions that we can find on the periodic table, magnesium and iron, And these types of elements sometimes are necessary for an enzyme to function. Okay, so they're not part of the protein, but they become part of that enzyme. All right, so good enough. Let's move on. So the next one is the one that I spent more time on, and this is this idea of how enzymes now get regulated. So regulation means what does the body do to ensure that enzymes are functioning properly, sometimes to turn on an enzyme so that it does something, sometimes to turn off an enzyme. And so let's take a look at some ways that regulation can take place. So there are things which we know are reversible, some regulators that are out there that can act to either turn on or turn off an enzyme. And so we're going to see here, a first example is something called a competitive inhibitor. So if this blue triangle here is representing a substrate for this particular enzyme, we know that typically in normal situations, if we have both enzyme and substrate, the substrate is going to enter this active site, and then the enzyme is going to do something with it. So it's going to help it to break down, or maybe it's going to help it to build something else. But a competitive inhibitor is a molecule that is similar in shape to the substrate, and therefore it can go into that active site and basically temporarily inhibit that enzyme from acting on a substrate. So it can go in and out of this active site, and it doesn't go in. Remember that we're talking about hundreds, if not thousands, of enzymes sometimes actively doing things in your body. So if this is in your digestive tract and we're talking about pepsin as the one that starts to break down proteins in your stomach, then if we had an inhibitor in there, that would mean that the pepsin would, at least temporarily, at least some of those enzymes would not be able to be as active because this inhibitor was in the way. But it's reversible, so this inhibitor will leave that active site, and then when it does, if a substrate is nearby, then the enzyme can act on it. So those are competitive inhibitors. The reason they're called competitive is they compete with the substrate for that enzyme's active site. Now there's also some things we call non-competitive, and those are ones that change the shape of the enzyme. So we call those allosteric because they cause the enzyme to form a different shape temporarily. So if it's an allosteric inhibitor, notice this little wedge over here, this is what I'm saying is the allosteric inhibitor for that enzyme. That means that it can enter into a site that we actually call the allosteric site, which is over here on this enzyme. And when it does that, what it's doing is changing the shape of this active site so that the substrate can't fit in. So that's a way of inhibiting by changing the shape. Allosteric inhibitor. Okay, still reversible though. There's also something called an allosteric activator. So in the case where an enzyme is normally in this inactive form, where the active area here, right, its active site isn't exposed, then it can get turned on by changing its shape through an allosteric activator. And so the activator can come in, locks into this site that we're saying is the allosteric site, and by doing that it opens up the enzyme's active site. So now this enzyme is active. Sometimes a phosphate will do that, so that's why there's little phosphates here. Sometimes putting a phosphate group on from ATP can cause it to activate an enzyme. Sometimes that's referred to as phosphorylation of something, is to activate it using one of those phosphates from ATP. But any case, what the same idea is that we're changing the shape of an enzyme. This time we're changing it so that it's favorable to pick up the substrate. So if that makes sense. Hopefully it does. So if we saw this on a graph, and you do have this on OpenStax, we can see that a normal enzyme, so we're not talking about any inhibitors or activators, well, we could be talking about an activator, but right now we're just talking about inhibitors. here you can see that a normal enzyme, this would be its activation rate when we had plenty of substrate. But if we have a competitive inhibitor, look what happens. It's a little bit lower activation, but ultimately it will get up to that normal level given enough substrate and in enough time. When you have a non-competitive inhibitor, though, the one that changes the shape of the enzyme, so sort of puts it out of commission a little bit, you're going to see even a lower rate of activity, and that doesn't reach the normal level. So these are just two different ways that inhibition can happen: competitively, or in this case, non-competitively, which would be that allosteric inhibition. The last thing on this sheet is this irreversible inhibitors. So there are certain things like toxins and poisons that can go into enzyme active sites and stay there, never leave. And those are actually really bad. We don't want to be around those because those truly lock up forever that enzyme. So therefore that enzyme is incapacitated, and that can even be fatal. So I'm not going to get into great detail on this. There's a lot of good examples I talked about in class when we were looking at insecticides, for example. So those would be things which could irreversibly inhibit some enzymatic activity. All right. Finally, we get to the last piece, and this is the metabolic pathways that we said. Well, in most cases in the body, metabolism is usually a multi-step process. And so I'll show you the relationship between Gibbs free energy equation and some of these steps in this process. So here's the example. We have substrate W that's going to get converted to a product X. Then product X becomes the substrate and gets converted to product Y. Then product Y becomes the substrate, and ultimately we get to what we really are looking for, and that's this final product Z. So each one of this is a multi-step reaction. Each one has its own enzyme, so I kind of put up here W-ACE is the enzyme for this one. I just drew a little picture here. Here is X-ACE, because that's the enzyme that catalyzes this reaction going from X to Y. And then Y-ACE being the reaction that catalyzes this one from Y to Z. So we get to the final product, and then we get a lot of product, we want to shut this pathway off. So one of the ways that happens in the body is that one of the products can go back and this would happen to multiple enzymes, not just a single enzyme, but it would go into this this allosteric site. It would change this enzyme. It would basically inhibit it. So it would be kind of like temporarily stopping this activity because we have plenty of this product. We don't need any more. Let's shut it off early right here in this first step of the pathway. That's feedback inhibition. Finally, let's look at this idea of Gibbs free energy. So this equation, you don't really need to worry too much about the equation at this point, but just looking at this thing called delta G. If you have a reaction that is a catabolic reaction, one that energy is going to get released and the product side of things, one that we would call exergonic, what you'll see is that delta G value is going to be less than 0. So I can judge a reaction by seeing what this delta G value is for that particular part of the reaction. If it's an endergonic reaction, that would be something we'd say energy is required actually in the reactant side in order to push this reaction forward. In that case, the delta G would be greater than 0. It would be a positive number, positive value. And I think, hold on one sec. Okay, just to finish that up just briefly, here is an actual reaction that we're about to get into and see how many steps it is to take glucose and to make a product here at the end. And you can see wherever there's energy added in, that one would be where a delta G would be positive. So that's it.